Hello and welcome to the Hospital Near Me Medical Career Training Channel. Today we are working on the Medical College Admissions Test, or MCAT, to get you into medical school. Today's featured hospital is Long Island Community Hospital, 631-654-71000, 101 Hospital Road, Patchogue, NY, 11772, lightcommunityhospital.org/. Welcome to our channel. Today, we're diving deep into the fundamental elements of genetics and microbiology. Join us as we explore the intricacies of DNA, RNA myths, ATP synthesis, bacterial spores, viruses, and more. In this educational video, we delve into the fascinating world of genetics and microbiology, learn about the foundational building blocks of DNA, debunk common myths about RNA, discover the chemical bonds that power ATP synthesis, explore bacterial spores, understand retroviruses and the Lytic Cycle of viruses, and explore genetic variation and antibiotic resistance in bacteria. Do you need to talk to someone? Get confidential help through talking with a full-services professional listening company. Reduce your stress and increase your peace. Get problem-solving help today. Click on the link in the description that says, "Let me help you in life." Increase your peace. Get problem-solving help today. Question 1. Building blocks of DNA. Discuss the structure of DNA and its essential components, nucleotides, sugar phosphate backbone. Explain how these building blocks form the double helix structure. Deoxyribonucleic acid, DNA, is a complex molecule that serves as the blueprint for life in organisms. Its structure consists of 4 types of nucleotides, which are the building blocks of DNA. Each nucleotide consists of 3 components: a phosphate group, a sugar molecule, deoxyribose in DNA, and a nitrogenous base. Phosphate group, PO4-screenase. The phosphate group is a key component of the DNA backbone. It forms a covalent bond with the 3 carbon of 1-deoxyribose sugar and the 5 carbon of the adjacent deoxyribose sugar, creating a sugar-phosphate backbone. The phosphate groups give DNA its negative charge, which is important for its interaction with other molecules in cellular processes, such as replication and transcription. Deoxyribose sugar. Deoxyribose is a 5-carbon sugar molecule that forms the backbone of DNA alongside the phosphate groups. It provides a structural framework for the DNA molecule. The carbon atoms in the deoxyribose sugar are numbered 1 through 5. The phosphate group attaches to the 5' carbon, and the nitrogenous base attaches to the 1' carbon. The absence of an oxygen atom at the 2' position distinguishes deoxyribose from ribose, which is found in RNA. Nitrogenous bases. There are 4 types of nitrogenous bases found in DNA: adenine A, cytosine C, guanine G, and thymine T. Adenine A and guanine G are purines, characterized by a double ring structure. Cytosine C and thymine T are pyrimidines, characterized by a single ring structure. The nitrogenous bases pair with each other through hydrogen bonds. Adenine pairs with thymine, AT, and cytosine pairs with guanine, CG. This complementary base pairing is crucial for DNA replication and stability. The sequence of nitrogenous bases along the DNA strand determines the genetic information encoded within the molecule, as the order of bases corresponds to specific genes and their functions. Understanding the structure and properties of DNA's building blocks is essential for comprehending fundamental biological processes such as DNA replication, transcription, and translation. Mastery of these concepts is crucial for success in exams such as the MCAT, Medical College Admission Test, as they form the basis for understanding genetics, molecular biology, and biochemistry. Question 2. Statement that is not true about RNA, debunking RNA myths. Address common misconceptions about RNA, e.g., it's just a messenger, not involved in regulation. Highlight the diverse functions of RNA beyond protein synthesis. Common misconceptions about RNA, debunking MCAT myths. RNA, ribonucleic acid, is a fundamental molecule essential for various biological processes within cells. Understanding RNA's structure, function, and properties is crucial for success in the Medical College Admission Test, MCAT. However, several misconceptions about RNA persist, potentially leading to incorrect answers on the exam. This article aims to address one statement that is not true about RNA, elucidating the correct understanding to aid MCAT preparation. Statement: RNA is only involved in protein synthesis. While RNA's role in protein synthesis, particularly through translation, is well known, this statement oversimplifies RNA's multifaceted functions within cells. RNA plays diverse roles beyond protein synthesis, contributing significantly to cellular processes and gene expression regulation. Below are key aspects to consider. mRNA, messenger RNA. mRNA carries genetic information from the DNA in the nucleus to the ribosomes in the cytoplasm, where it serves as a template for protein synthesis during translation. This process is central to the flow of genetic information, but it represents only one of the many functions of RNA. transfer RNA. tRNA molecules deliver amino acids to the ribosome during protein synthesis. These molecules possess anticodons that base pair with complementary codons on mRNA, ensuring the accurate incorporation of amino acids into the growing polypeptide chain. Despite their primary role in translation, tRNAs are also involved in other cellular processes, such as RNA splicing and RNA editing. rRNA, ribosomal RNA. rRNA is a structural component of ribosomes, the cellular organelles responsible for protein synthesis. Ribosomes consist of both protein and rRNA molecules, forming the machinery where mRNA is translated into protein. Beyond protein synthesis, rRNA also plays a role in the catalytic activity of the ribosome during peptide bond formation. Regulatory RNAs. Various classes of regulatory RNAs, such as microRNAs, miRNAs, and long non-coding RNAs, lncRNAs, modulate gene expression at the transcriptional or post-transcriptional level. These RNAs can interact with mRNA molecules, leading to their degradation or inhibiting their translation. Additionally, regulatory RNAs participate in processes like chromatin remodeling and epigenetic regulation, exerting profound effects on cellular function and development. Contrary to the statement that RNA is solely involved in protein synthesis, RNA exhibits diverse functions crucial for cellular processes and gene regulation. Understanding RNA's multifaceted roles beyond translation is essential for success on the MCAT and for comprehensively grasping cellular biology. Aspirants should familiarize themselves with the various types of RNA and their respective functions to excel in MCAT questions related to molecular biology and genetics. Chemical bonds that convert energy to ATP play a critical role in cellular energy metabolism, a topic commonly tested on the MCAT, Medical College Admission Test. Here's a college-level overview of this concept. 1. ATP, the universal energy currency. Adenosine triphosphate, ATP, is often referred to as the energy currency of the cell because it provides the energy necessary for cellular processes. ATP consists of 3 phosphate groups attached to an adenosine molecule. The bonds between these phosphate groups are high-energy bonds. 2. Energy conversion. Cellular processes, such as muscle contraction, active transport, and biosynthesis, require energy. This energy is derived from the breakdown of ATP. When ATP is hydrolyzed, meaning it reacts with water, it releases energy by breaking the high-energy bond between the last phosphate group and forming adenosine diphosphate, ADP, and inorganic phosphate, pi. 3. Chemical bonds involved. The conversion of ATP to ADP and pi involves the cleavage of the terminal phosphate bond, which is a high-energy bond. The energy released during this hydrolysis reaction is around 30.6 kilos per mole, which is utilized by the cell to drive various biochemical reactions. Number 4. Enzymes and ATP synthesis. The synthesis of ATP from ADP, known as ATP synthesis, occurs primarily in the mitochondria through a process called oxidative phosphorylation. Enzymes known as ATP synthases facilitate this process by coupling the synthesis of ATP to the flow of proton H+ across the mitochondrial inner membrane, creating a proton gradient. The energy released from the movement of protons down their electrochemical gradient drives the synthesis of ATP from ADP and pi. Number 5. Importance in cellular functions. ATP is crucial for the functioning of various cellular processes, including metabolism, DNA replication, muscle contraction, and nerve impulse transmission. Without ATP, these processes would cease, leading to cellular dysfunction and ultimately organismal death. 6. Biological significance. Understanding the mechanisms of ATP synthesis and hydrolysis is essential for grasping the fundamentals of cellular energetics and metabolism. Dysfunction in ATP production or utilization can lead to various pathological conditions such as metabolic disorders, muscular dystrophies, and neurodegenerative diseases. In summary, the chemical bonds involved in the conversion of energy to ATP, particularly in ATP hydrolysis and synthesis, are fundamental concepts tested on the MCAT. Mastery of these concepts is essential for understanding cellular energetics and metabolism, as well as their implications for human health and disease. Bacterial spores, a comprehensive overview. Bacterial spores are a remarkable survival strategy employed by certain species of bacteria, enabling them to withstand adverse environmental conditions such as extreme temperatures, desiccation, and exposure to chemicals or radiation. These spores are highly resistant structures formed by a process called sporulation or sporogenesis, which is triggered by unfavorable growth conditions. Understanding the formation, structure, and significance of bacterial spores is essential in various fields including microbiology, medicine, and biotechnology. This knowledge is often tested in standardized exams like the Medical College Admission Test, MCAT. Here's an overview of key points. 1. Sporulation process. Sporulation begins when bacterial growth conditions become unfavorable, triggering a cascade of genetic and biochemical events. The process involves the transformation of a vegetative cell into a spore, which is metabolically inactive and highly resistant to environmental stressors. Key stages in sporulation include initiation, asymmetric septation, engulfment, cortex formation, coat synthesis, spore maturation, and sporangium lysis. 2. Structure of bacterial spores. Bacterial spores exhibit a complex structure with multiple layers that provide protection against harsh environmental conditions. The core of the spore contains the bacterial chromosome, ribosomes, and enzymes required for germination. Surrounding the core is the cortex, a thick layer of peptidoglycan that provides structural integrity. The cortex is enclosed by the spore coat, a proteinaceous layer that further enhances resistance to environmental stressors. Some spores also possess additional layers such as exosporium, which further protects the spore from external threats. 3. Germination and spore revival. Under favorable conditions, bacterial spores can undergo germination, a process in which they return to the vegetative state. Germination involves the activation of specific germination receptors on the spore surface, triggering biochemical changes that lead to spore swelling, rupture of the spore coat, and resumption of metabolic activity. Once germinated, the spore can resume growth and proliferation, initiating a new vegetative cell cycle. 4. Clinical significance and applications. Bacterial spores play a significant role in the transmission and persistence of infectious diseases, as they can survive in the environment for extended periods and resist common disinfection methods. Certain pathogenic bacteria, such as Clostridium difficile and Bacillus anthracis, produce spores that contribute to their virulence and ability to cause disease. Understanding the mechanisms of sporulation and germination is crucial for developing strategies to prevent and control bacterial infections. Bacterial spores also have applications in biotechnology, such as the production of enzymes, antibiotics, and vaccines, as well as in food preservation and sterilization processes. 5. Research and future directions. Ongoing research in microbiology aims to elucidate the molecular mechanisms underlying sporulation and germination, with the goal of developing novel antimicrobial strategies. Advances in genetic engineering and synthetic biology may enable the manipulation of sporulation pathways for biotechnological applications and the development of novel therapeutics. Studying bacterial spores provides insights into microbial ecology, evolution, and adaptation to changing environments, contributing to our understanding of microbial communities and their interactions. In summary, bacterial spores are specialized structures that enable certain bacteria to survive adverse conditions and persist in the environment. Understanding the formation, structure, and significance of bacterial spores is essential for various fields, including medicine, microbiology, and biotechnology, and is a topic commonly tested on exams such as the MCAT.
Question 5. Retroviruses. Retroviruses and the Lytic Cycle. Introduce retroviruses and their unique replication strategy. Explain the lytic cycle of viruses, focusing on attachment, penetration, replication, assembly, and release. Retroviruses, a college-level overview. Retroviruses are a group of RNA viruses characterized by their ability to reverse transcribe their RNA genome into DNA upon infection of a host cell. This unique characteristic distinguishes them from other types of viruses and has significant implications for their replication, pathogenesis, and treatment. Understanding retroviruses is crucial for medical students preparing for the MCAT, as they play a vital role in various diseases and therapeutic approaches. Genome structure and replication. Retroviruses possess a single-stranded RNA genome enclosed within a protein capsid. The genome contains three main genes: GAG, POL, and NV. Upon infection, retroviruses use the enzyme reverse transcriptase to transcribe their RNA genome into double-stranded DNA. This DNA integrates into the host cell's genome, becoming a provirus. The integrated provirus can remain latent or become transcriptionally active, leading to the production of viral RNA and proteins. Examples and associated diseases. Human immunodeficiency virus, HIV, is the most well-known retrovirus, causing acquired immunodeficiency syndrome. HIV primarily targets CD4 plus T cells, leading to immune deficiency and susceptibility to opportunistic infections. Human T-cell leukemia virus type 1, HTLV1, is another retrovirus associated with diseases such as adult T-cell leukemia lymphoma and HTLV1-associated myelopathy, tropical spastic paraparesis. Treatment and therapeutic strategies. Antiretroviral therapy, ART, is the primary treatment for retroviral infections, particularly HIV. ART involves the use of a combination of drugs targeting different stages of the retroviral life cycle, such as reverse transcriptase inhibitors, protease inhibitors, and integrase inhibitors. Highly active antiretroviral therapy, ART, also known as combination therapy, has significantly improved the prognosis and quality of life for individuals with HIV/AIDS. Novel therapeutic approaches, including gene editing technologies like CRISPR-Cas9, hold promise for targeting and potentially eradicating retroviral infections by disrupting viral DNA integration or enhancing host immune responses. Viral evolution and drug resistance. Retroviruses, particularly HIV, exhibit high mutation rates due to the error-prone nature of reverse transcriptase and the lack of proofreading mechanisms. This rapid mutation can lead to the emergence of drug-resistant viral strains. Drug resistance poses a significant challenge in the management of retroviral infections, necessitating the development of new antiretroviral drugs and treatment strategies. Public health and prevention. Prevention strategies for retroviral infections primarily focus on education, promotion of safe sex practices, access to HIV testing and counseling, needle exchange programs for intravenous drug users, and pre-exposure prophylaxis (PrEP) for high-risk individuals. Public health initiatives, such as awareness campaigns, community outreach, and access to healthcare services, are essential for reducing the transmission and burden of retroviral diseases globally. Conclusion. Retroviruses are a diverse group of RNA viruses with unique replication strategies and significant implications for human health. Understanding their biology, associated diseases, treatment options, and prevention strategies is essential for medical students preparing for the MCAT and for healthcare professionals involved in the diagnosis and management of retroviral infections. Question 6. Viruses' lytic cycle. Explain the lytic cycle of viruses, focusing on attachment, penetration, replication, assembly, and release. Viruses, lytic cycle. Viruses are microscopic infectious agents that can only replicate within the living cells of organisms. The lytic cycle is one of the two main replication cycles employed by viruses, the other being the lysogenic cycle. Understanding the lytic cycle is crucial for grasping the mechanisms of viral replication and its implications in diseases. Attachment. The lytic cycle begins when a virus attaches itself to a specific receptor site on the surface of a host cell. This attachment is highly specific and is mediated by viral surface proteins interacting with complementary receptor molecules on the host cell. Penetration. Once attached, the virus injects its genetic material into the host cell. This genetic material could be either DNA or RNA, depending on the type of virus. Once inside the host cell, the virus utilizes the cellular machinery to replicate itself. Biosynthesis. After penetration, the viral genetic material hijacks the host cell's machinery, directing it to produce viral components such as nucleic acids, proteins, and structural components. Viral enzymes often manipulate the host cell's metabolic pathways to favor viral replication. Maturation. During this stage, newly synthesized viral components assemble to form complete viral particles called virions. This process usually occurs in the cytoplasm of the host cell. Lysis and release. Once assembled, the mature virions break open, lyse, the host cell, causing it to burst and release a large number of viral particles into the surrounding environment. These released virions can then infect neighboring cells and continue the Lytic cycle. Significance and MCAT relevance. Understanding the Lytic cycle is crucial for medical professionals, especially in the context of viral diseases. For example, many acute viral infections, such as influenza and the common cold, follow the Lytic cycle, leading to the rapid onset of symptoms. On the MCAT, questions related to the Lytic cycle may assess a test taker's understanding of viral replication, host pathogen interactions, and the potential targets for antiviral therapies. Additionally, knowledge of the Lytic cycle contributes to understanding the spread and control of viral infections, which is essential for public health measures and disease management strategies. In summary, the Lytic cycle is a fundamental process in viral replication, wherein viruses hijack host cell machinery to produce new viral particles, leading to cell lysis and the release of progeny virions. Understanding this cycle is essential for comprehending viral pathogenesis and designing effective therapeutic interventions.
The lytic cycle is a key process in viral replication within bacteria. It involves several distinct steps: attachment, penetration, biosynthesis, maturation, and lysis. Each step plays a crucial role in the virus's ability to hijack bacterial machinery for reproduction. The lytic cycle is a fundamental process employed by bacteriophages, or viruses that infect bacteria, to replicate and propagate themselves. Understanding the order of steps in the lytic cycle is crucial in comprehending viral replication mechanisms, which is a common topic in biology and microbiology curricula, as well as standardized tests like the MCAT, Medical College Admission Test. Attachment. The first step in the lytic cycle involves the attachment of the bacteriophage to the surface of the bacterial cell. This attachment is mediated by specific binding interactions between viral proteins, such as tail fibers or spikes, and receptors on the bacterial cell wall. This step is critical for ensuring specificity in viral infection, as different bacteriophages may have different receptor-binding proteins that recognize distinct receptors on the bacterial surface. Penetration. After attachment, the bacteriophage injects its genetic material, usually DNA, but sometimes RNA, into the host bacterial cell. This process often involves the contraction of the phage tail sheath, which acts like a syringe, puncturing the bacterial cell wall and delivering the viral genome into the cytoplasm of the host cell. Once inside the host cell, the viral genetic material becomes free to exploit the host's cellular machinery for replication. Biosynthesis. With the viral genetic material now inside the host cell, the next step is the biosynthesis of viral components. The host cell's machinery is hijacked to transcribe and translate viral genes, leading to the production of viral proteins and replication of viral nucleic acids. Viral DNA replication typically occurs using the host's DNA polymerase enzyme, while viral proteins are synthesized using the host's ribosomes. As a result, new viral particles begin to assemble within the host cell. Maturation. As the newly synthesized viral components accumulate within the host cell, they undergo assembly to form complete viral particles. This process often involves the self-assembly of viral capsids around the replicated viral nucleic acids, as well as the packaging of viral enzymes and other structural proteins. Once assembly is complete, the maturation of viral particles may involve additional processing steps, such as proteolytic cleavage of precursor proteins or the addition of viral envelopes derived from the host cell membrane. Release. The final step in the lytic cycle is the release of newly formed viral particles from the host cell. This typically occurs through the lysis, or bursting, of the host cell, which is triggered by the accumulation of viral particles and the expression of viral proteins that disrupt the host cell membrane. Once released, the mature viral particles can infect new host cells and initiate subsequent rounds of infection, thereby propagating the lytic cycle. Understanding the order of these steps in the lytic cycle is essential for comprehending viral replication strategies and host pathogen interactions, which are foundational concepts in microbiology and virology, as well as in medical fields such as immunology and infectious diseases. Consequently, knowledge of the lytic cycle and its stages is frequently tested on standardized exams like the MCAT, which assesses students' understanding of biological concepts relevant to medical education and practice. Now let's talk about plasmids. These small, circular DNA molecules can carry extra genes that provide advantages to bacteria, such as antibiotic resistance. Plasmids can be transferred between bacterial cells, facilitating the spread of resistance genes. Genetic material is the blueprint of life, encoding the instructions necessary for the growth, development, and functioning of all living organisms. In cellular organisms, genetic material exists in two primary forms: plasmids and chromosomes. Understanding the distinctions and interactions between these two entities is fundamental to comprehending various biological processes, including gene expression, replication, and genetic engineering. Plasmids are small, circular, extrachromosomal DNA molecules commonly found in bacteria, though they can also exist in other types of cells. Unlike chromosomes, which typically contain the majority of an organism's genetic information, plasmids are relatively small and may carry only a few genes. These genes often encode traits that provide selective advantages to the host organism under specific environmental conditions, such as antibiotic resistance or the ability to metabolize certain compounds. Features of plasmids: autonomous replication. Plasmids can replicate independently of the host chromosome. They possess their own origin of replication, ORI, allowing them to initiate synthesis and propagate within the host cell. Variable size and content. Plasmids can vary greatly in size, ranging from a few thousand to several 100,000 base pairs. Additionally, their genetic content is diverse and can include genes for antibiotic resistance, virulence factors, metabolic enzymes, and other functions. Conjugation and horizontal gene transfer. Plasmids often carry genes that facilitate their transfer between bacterial cells through processes like conjugation. This horizontal gene transfer allows bacteria to acquire new traits rapidly, contributing to their adaptability and evolution. The transfer of genetic material from plasmids to chromosomes, known as integration, can occur through various mechanisms. One common method involves the integration of plasmid DNA into the host chromosome via recombination events mediated by specialized enzymes such as integrases. This integration can lead to stable inheritance of the plasmid-encoded traits by the host cell's progeny. Understanding the dynamics between plasmids and chromosomes has significant implications for both basic research and applied biotechnology. For instance, the study of plasmid-chromosome interactions elucidates mechanisms of gene regulation, genetic recombination, and evolutionary adaptation in bacteria. Moreover, the ability to manipulate plasmid DNA and facilitate its integration into host chromosomes forms the basis of various biotechnological tools, such as gene cloning, recombinant DNA technology, and gene therapy. In summary, plasmids and chromosomes represent distinct but interconnected components of the genetic material in cellular organisms. While chromosomes serve as the primary repository of genetic information, plasmids play essential roles in facilitating horizontal gene transfer, promoting genetic diversity, and driving evolutionary innovation. Understanding the mechanisms underlying the transfer of genetic material between plasmids and chromosomes is crucial for advancing our knowledge of cellular biology and harnessing biotechnological applications.
Genetic variation in bacteria plays a crucial role in their adaptation to changing environments, antibiotic resistance, and evolution. Several mechanisms contribute to genetic variation in bacterial populations, including mutation, horizontal gene transfer, and recombination. Mutation. Mutations are changes in the DNA sequence of an organism. Bacteria can acquire mutations through errors in DNA replication, exposure to mutagens such as UV radiation or certain chemicals, or through the action of mobile genetic elements like transposons. Mutations can lead to variations in bacterial traits, including antibiotic resistance, metabolic capabilities, and virulence. Horizontal gene transfer, HGT. Unlike vertical gene transfer, which occurs from parent to offspring, HGT involves the transfer of genetic material between different bacterial cells. HGT mechanisms include transformation, transduction, and conjugation. Transformation. Bacteria can take up free DNA from their environment and incorporate it into their own genome. This process is facilitated by specialized proteins on the bacterial cell surface. Transduction. Bacteriophages, viruses that infect bacteria, can transfer bacterial DNA from one cell to another during the infection process. This can result in the transfer of genes between different bacterial species. Conjugation. In conjugation, genetic material is transferred between bacterial cells through direct cell-to-cell contact. This transfer typically involves plasmids, which are small, circular DNA molecules separate from the bacterial chromosome. Conjugation can facilitate the spread of antibiotic resistance genes and other advantageous traits among bacterial populations. Recombination. Recombination is the process by which genetic material from two different sources combines to form new combinations of genes. In bacteria, recombination can occur between homologous regions of DNA during processes such as transformation, transduction, or conjugation. Recombination contributes to genetic diversity by shuffling genetic material between bacterial genomes. Understanding genetic variation in bacteria is crucial for various fields, including medicine, agriculture, and environmental science. For example, in the medical field, knowledge of bacterial genetic variation informs strategies for combating antibiotic resistance and developing new antimicrobial agents. In agriculture, understanding bacterial genetic variation can help in the development of more effective strategies for controlling plant diseases and promoting beneficial interactions between bacteria and crops. Additionally, in environmental science, studying bacterial genetic variation can provide insights into microbial diversity and ecosystem function. Antibiotic resistance is a growing concern worldwide. Bacteria can acquire resistance through mutations in their own DNA or by acquiring resistance genes from plasmids. This adaptation allows them to survive and thrive in the presence of antibiotics. Understanding these processes is crucial for developing strategies to combat antibiotic resistance and to appreciate the intricate world of bacterial evolution. Antibiotic resistance is a significant global health concern resulting from the ability of bacteria to adapt and survive in the presence of antibiotics. This phenomenon occurs through various mechanisms, including genetic mutations, horizontal gene transfer, and selective pressure. Genetic mutations. Bacteria can acquire resistance to antibiotics through spontaneous mutations in their genetic material. These mutations can alter the structure or function of antibiotic targets, such as enzymes involved in cell wall synthesis or DNA replication. Mutations may lead to reduced antibiotic binding affinity, rendering the antibiotic ineffective in inhibiting bacterial growth or killing the bacteria. Horizontal gene transfer, HGT. Bacteria can transfer genetic material horizontally between different strains or species, leading to the spread of antibiotic resistance genes. Common mechanisms of HGT include conjugation, transformation, and transduction. Conjugation involves the transfer of genetic material through direct cell-to-cell contact via conjugative pilus. Transformation involves the uptake of naked DNA from the environment, while transduction involves the transfer of genetic material via bacteriophages, viruses that infect bacteria. Selective pressure. The widespread use and misuse of antibiotics exerts selective pressure on bacterial populations, favoring the survival and proliferation of resistant strains. Exposure to sublethal concentrations of antibiotics can promote the emergence of resistant bacteria by providing an environment where only resistant mutants can survive and reproduce. Additionally, incomplete antibiotic courses or inappropriate antibiotic prescriptions can contribute to the development of resistance by allowing surviving bacteria to proliferate and spread. Antibiotic resistance mechanisms. Bacteria employ various mechanisms to resist the actions of antibiotics, including enzymatic inactivation of antibiotics. Bacteria may produce enzymes that chemically modify or degrade antibiotics, rendering them inactive. Examples include beta-lactamases, which hydrolyze beta-lactam antibiotics like penicillins and cephalosporins. Alteration of antibiotic targets. Bacteria can modify the structure or expression of antibiotic targets, such as ribosomes or cell wall components, preventing antibiotics from binding effectively. Efflux pumps. Bacteria may possess efflux pumps that actively remove antibiotics from the intracellular environment, reducing their concentration within the bacterial cell and conferring resistance. Clinical implications. Antibiotic resistance poses a significant challenge in the treatment of bacterial infections, leading to treatment failures, prolonged illnesses, and increased healthcare costs. Multi-drug-resistant bacteria, such as methicillin-resistant Staphylococcus aureus, MRSA, and carbapenem-resistant Enterobacteriaceae, CRE, have become increasingly prevalent in healthcare settings, limiting treatment options and posing serious public health threats. Addressing antibiotic resistance requires a multifaceted approach, including judicious antibiotic use, development of novel antibiotics, infection prevention and control measures, and public education on the responsible use of antibiotics. Understanding the mechanisms and implications of antibiotic resistance is crucial for healthcare professionals in effectively managing bacterial infections and combating the spread of resistant bacteria.
Question 1. Building blocks of DNA. Which of the following statements accurately describes the building blocks of DNA? A DNA is composed of nucleotides, each consisting of a phosphate group, a sugar molecule, and a nitrogenous base. B. DNA is made up of amino acids, each containing a phosphate group, a sugar molecule, and a nitrogenous base. C. DNA consists of amino acids, each consisting of a phosphate group, a ribose sugar molecule, and a nitrogenous base. D. DNA is comprised of nucleotides, each containing a phosphate group, a ribose sugar molecule, and a nitrogenous base. Question 2. Statement that is not true about RNA. Which of the following statements is not true about RNA? A. RNA is typically single-stranded. B. RNA contains the nitrogenous base uracil. C. RNA serves primarily as a long-term storage molecule in cells. D. RNA is involved in protein synthesis. Question 3. Chemical bonds that convert energy to ATP. Which chemical bond play a key role in converting energy to ATP during cellular respiration? A. Covalent bonds in glucose molecules. B. Ionic bonds in water molecules. C. Hydrogen bonds in DNA strands. D. Phosphoanhydride bonds in ATP molecules. Question 4. Bacterial spores. Which of the following statements regarding bacterial spores is correct? A. Bacterial spores are formed as a response to favorable environmental conditions. B. Bacterial spores are primarily produced by gram-negative bacteria. C. Bacterial spores are reproductive structures involved in bacterial cell division. D. Bacterial spores are highly resistant structures formed in response to adverse environmental conditions. Question 5. Retroviruses. Which of the following statements accurately describes a characteristic feature of retroviruses? A. Retroviruses replicate through a DNA intermediate using reverse transcriptase. B. Retroviruses contain single-stranded RNA as their genetic material. C. Retroviruses primarily infect prokaryotic cells. D. Retroviruses are unable to integrate their genome into the host cell's DNA. Question 6. Viruses' Lytic Cycle. Which of the following accurately describes a key event in the Lytic cycle of viruses? A. Integration of viral DNA into the host genome. B. Release of mature virions from the host cell. C. Production of viral proteins by the host cell machinery. D. Formation of a prophage within the host cell. Question 7. The order of the steps in the Lytic cycle. Which of the following represents the correct order of steps in the Lytic cycle? A. Penetration, replication, maturation, lysis, attachment. B. Attachment, penetration, maturation, replication, lysis. C. Attachment, maturation, penetration, replication, lysis. D. Maturation, attachment, penetration, replication, lysis. Question 8. Genetic material from the plasmid. Which of the following statements accurately describes the transfer of genetic material from a plasmid to the chromosome? A. Plasmid DNA integrates into the chromosome through a process called transformation. B. Plasmid DNA is transferred to the chromosome via conjugation. C. Plasmid DNA integrates into the chromosome through transduction. D. Plasmid DNA is directly incorporated into the chromosome through transfection. Question 9. Genetic variation in bacteria. Which of the following mechanisms contributes significantly to genetic variation in bacteria? A. Conjugation. B. Mitosis. C. Meiosis. D. Binary fission. Question 10. Antibiotic resistance of bacterial population. Which of the following scenarios would most likely lead to the rapid emergence of antibiotic resistance in a bacterial population? A. Infrequent use of antibiotics in a community setting. B. Consistent and proper use of antibiotics as prescribed by healthcare providers. C. Overuse and misuse of antibiotics in agriculture and livestock farming. D. Limited availability of antibiotics in low-income countries. Question 1. Building blocks of DNA. Which of the following statements accurately describes the building blocks of DNA? A. DNA is composed of nucleotides, each consisting of a phosphate group, a sugar molecule, and a nitrogenous base. B. DNA is made up of amino acids, each containing a phosphate group, a sugar molecule, and a nitrogenous base. C. DNA consists of amino acids, each consisting of a phosphate group, a ribose sugar molecule, and a nitrogenous base. D. DNA is comprised of nucleotides, each containing a phosphate group, a ribose sugar molecule, and a nitrogenous base. Correct answer: A. DNA is composed of nucleotides, each consisting of a phosphate group, a sugar molecule, and a nitrogenous base. Explanation: this statement correctly describes the structure of DNA. Nucleotides are indeed the building blocks of DNA, and each nucleotide consists of a phosphate group, a sugar molecule, deoxyribose in the case of DNA, and a nitrogenous base, adenine, thymine, cytosine, or guanine. B. This statement is incorrect. Amino acids are not the building blocks of DNA. Rather, they are the building blocks of proteins. Additionally, the components listed, phosphate group, sugar molecule, and nitrogenous base, are characteristic of nucleotides, not amino acids. C. This statement is incorrect. Amino acids are not the constituents of DNA. They are the building blocks of proteins. Furthermore, DNA contains deoxyribose sugar, not ribose sugar as stated in this option. D. This statement is incorrect. While DNA is indeed composed of nucleotides, the sugar molecule in DNA is deoxyribose, not ribose as mentioned here. Additionally, amino acids are not part of the structure of DNA. They are the building blocks of proteins. Question 2. Statement that is not true about RNA. Which of the following statements is not true about RNA? A. RNA is typically single-stranded. B. RNA contains the nitrogenous base uracil. C. RNA serves primarily as a long-term storage molecule in cells. D. RNA is involved in protein synthesis. Correct answer: C. RNA serves primarily as a long-term storage molecule in cells. Explanation: this statement is true. RNA molecules are typically single-stranded, unlike DNA, which is double-stranded. B. This statement is true. RNA contains the nitrogenous base uracil instead of thymine, which is found in DNA. C. This statement is not true. RNA does not serve primarily as a long-term storage molecule in cells. This function is primarily attributed to DNA, which serves as the repository of genetic information in cells. RNA, on the other hand, is involved in various cellular processes including protein synthesis, gene regulation, and catalysis. D. This statement is true. RNA plays a crucial role in protein synthesis through processes like transcription and translation. In summary, option C is the correct answer because RNA primarily serves as a functional molecule in cellular processes rather than as a long-term storage molecule. Options A, B, and D correctly describe characteristics and functions of RNA.
Question 3. Chemical bonds that convert energy to ATP. Which chemical bonds play a key role in converting energy to ATP during cellular respiration? A. Covalent bonds in glucose molecules. B. Ionic bonds in water molecules. C. Hydrogen bonds in DNA strands. D. Phosphoanhydride bonds in ATP molecules. Correct answer: D. Phosphoanhydride bonds in ATP molecules. Explanation: A. Covalent bonds in glucose molecules. While glucose molecules do undergo covalent bond breakage during cellular respiration, the energy released from these bonds is not directly used to produce ATP. Instead, the breakdown of glucose serves as a source of electrons that eventually contribute to the formation of ATP through oxidative phosphorylation. B. Ionic bonds in water molecules. Ionic bonds in water molecules are important for its structure and properties, but they are not directly involved in the conversion of energy to ATP during cellular respiration. C. Hydrogen bonds in DNA strands. Hydrogen bonds in DNA are essential for maintaining the double helix structure, but they are not directly involved in the process of converting energy to ATP. D. Phosphoanhydride bonds in ATP molecules. ATP, adenosine triphosphate, is the primary molecule used by cells to store and transfer energy. The conversion of ATP to ADP, adenosine diphosphate, involves the hydrolysis of a phosphoanhydride bond, releasing energy that can be used for cellular work. Therefore, phosphoanhydride bonds in ATP molecules are directly involved in the process of converting energy to ATP during cellular respiration. In summary, option D is the correct answer because phosphoanhydride bonds in ATP molecules directly participate in the conversion of energy to ATP during cellular respiration, whereas the other options are not directly involved in this process. Question 4. Bacterial spores. Which of the following statements regarding bacterial spores is correct? A. Bacterial spores are formed as a response to favorable environmental conditions. B. Bacterial spores are primarily produced by gram-negative bacteria. C. Bacterial spores are reproductive structures involved in bacterial cell division. D. Bacterial spores are highly resistant structures formed in response to adverse environmental conditions. Correct answer, D. Bacterial spores are highly resistant structures formed in response to adverse environmental conditions. Explanation. A. Incorrect. Bacterial spores are not formed in response to favorable environmental conditions. Rather, they are formed in response to adverse conditions such as nutrient depletion or exposure to extreme temperatures or chemicals. Spore formation is a survival mechanism rather than a response to favorable conditions. B. Incorrect. Bacterial spores are primarily produced by gram-positive bacteria, not gram-negative bacteria. Examples of gram-positive spore-forming bacteria include Bacillus and Clostridium species. C. Incorrect. Bacterial spores are not reproductive structures involved in bacterial cell division. Spores are dormant structures formed by certain bacterial species as a means of survival under unfavorable conditions. They are not involved in the reproduction process. Instead, they allow bacteria to survive harsh environments until conditions become favorable for growth and reproduction. D. Correct. Bacterial spores are indeed highly resistant structures formed in response to adverse environmental conditions. Spore formation, known as sporulation, is a protective mechanism employed by certain bacterial species to survive harsh conditions such as nutrient deprivation, extreme temperatures, or exposure to chemicals. Spores are highly resistant to heat, desiccation, and disinfectants, allowing bacteria to remain dormant until conditions improve. This resilience contributes to the survival and spread of spore-forming bacteria in various environments. Question 5. Retroviruses. Which of the following statements accurately describes a characteristic feature of retroviruses? A. Retroviruses replicate through a DNA intermediate using reverse transcriptase. B. Retroviruses contain single-stranded RNA as their genetic material. C. Retroviruses primarily infect prokaryotic cells. D. Retroviruses are unable to integrate their genome into the host cell's DNA. Correct answer. A. Retroviruses replicate through a DNA intermediate using reverse transcriptase. Explanation. Correct. Retroviruses are unique among viruses in that they replicate through a DNA intermediate. Upon infecting a host cell, the retroviral RNA genome is reverse transcribed into DNA by the enzyme reverse transcriptase. This DNA is then integrated into the host cell's genome, where it can be transcribed and translated to produce new viral particles. B. Incorrect. Retroviruses contain single-stranded RNA as their genetic material, not DNA. However, during replication, this RNA is converted into DNA through the action of reverse transcriptase. C. Incorrect. Retroviruses primarily infect eukaryotic cells, not prokaryotic cells. Their replication cycle involves integration into the host cell's DNA, which is a feature exclusive to eukaryotic cells. D. Incorrect. Retroviruses are capable of integrating their genome into the host cell's DNA. This integration is a crucial step in their replication cycle and is facilitated by the enzyme integrase. Question 6. Virus's Lytic Cycle. Which of the following accurately describes a key event in the Lytic cycle of viruses? A. Integration of viral DNA into the host genome. B. Release of mature virions from the host cell. C. Production of viral proteins by the host cell machinery. D. Formation of a prophage within the host cell. Correct answer B. Release of mature virions from the host cell. Explanation. Integration of viral DNA into the host genome is a characteristic of the lysogenic cycle, not the lytic cycle. During the lysogenic cycle, viral DNA becomes integrated into the host genome as a prophage. B. Release of mature virions from the host cell is a key event in the lytic cycle. After the viral replication and assembly within the host cell, mature virions are released through lysis, destroying the host cell in the process. C. Production of viral proteins by the host cell machinery occurs in both the lytic and lysogenic cycles. However, it is not a defining characteristic of the lytic cycle. D. Formation of a prophage within the host cell is a characteristic of the lysogenic cycle, not the lytic cycle. In the lysogenic cycle, the viral DNA becomes integrated into the host genome as a prophage without causing immediate destruction of the host cell. In summary, the correct answer is B because the release of mature virions from the host cell is a hallmark of the lytic cycle, distinguishing it from the lysogenic cycle. Choices A, C, and D are incorrect because they describe events associated with the lysogenic cycle. Question 7. The order of the steps in the lytic cycle. Which of the following represents the correct order of steps in the lytic cycle? A. Penetration, replication, maturation, lysis, attachment. B. Attachment, penetration, maturation, replication, lysis. C. Attachment, maturation, penetration, replication, lysis. D. Maturation, attachment, penetration, replication, lysis. Correct answer, B. Attachment, penetration, maturation, replication, lysis. Explanation. A. Incorrect. This option lists penetration as the first step, which is inaccurate. The first step in the lytic cycle is attachment, where the phage attaches to the host cell's surface receptor. C. Incorrect. This option incorrectly places maturation before penetration. Maturation occurs after replication, not before penetration. D. Incorrect. This option lists maturation as the initial step, which is incorrect. Maturation occurs later in the cycle after replication. B. Correct. This option correctly lists the steps in the lytic cycle in the following order. Attachment. The phage attaches to the host cell's surface. Penetration. The phage injects its genetic material into the host cell. Maturation. Viral components are synthesized and assembled. Replication. The viral genetic material is replicated and new viral particles are synthesized. Lysis. The host cell ruptures, releasing newly formed viral particles.